Perks to attend the oral presentations. Abstracts will be published in the conference proceeding with an ISBN numbers, gain SCRMP magazine coverage, second issue with ISSN numbers, chance to present your work with oral presentation and win exciting rewards. Bharat, change the slide, please. Judging criteria and scoring, how your presentation will be scored. First of all, content and relevancy will content about 30% of your marks. Alignment with the theme, accuracy, originality, and depth of the knowledge are some of the scoring criteria. Second one, clarity and organization. It will contain 20% of your marks. Logical structure, key points will be main criteria. Third one, delivery and communication. How good you can deliver and communicate with the judges will contain about 20% of your marks. Confidence, engagement, articulation, voice modulation are some of the criteria. Fourth one, visual aids and designs. Readability, visuals, professional layout are the criteria. Fifth, question and answer session. You will be having a viva for 2 to 3 minutes from the judges, and how you tackle the question would be a criteria for scoring. Next one. Final score is the average of all the judges' ratings. Our esteemed judging panel, first of all, Mr. Rahul Pal sir, he is an assistant professor at Department of Pharmaceutics in ISF College of Pharmacy, Moga. Second one, Dr. Payal, she is an MBBS PGD CR, Global Patient Safety, National Joint President at SCRMP, physician, certified psychotherapist and hypnotherapist. Third, Mr. Mohammad Gayur Khan, entrepreneur, General Secretary at SCRMP, ex-cognizant pharmacovigilance professional author, scientific writer, RWE, HUR. Mr. Shrikant, MD, MSc, MPM, and drug developer, physician scientist, and entrepreneur. Ms. Aishwarya Jain, pharmaceutical scientist, editor for Scopus Index Journals, innovator in nanotechnology, green synthesis. Bharat. Presentations timing rules are strict time limit for each speaker. Each participant has 10 minutes. Exceeding the time limits results in score deduction. Consequences of violating time rules. Warning will be given at 9th minute of the presentation, meaning before 10 minutes, 1 minute, it will be given a warning. Over 30 seconds over means at 10:30 penalty deductions, and more than 1 minute over means 11th minute possible disqualification. Speaker order and encouragement. Each participant will be called by name. We wish all the participants the best of luck. Top 3 winners will receive winner certificates, social media recognition, SCRMP magazine coverage, and exciting rewards.
Healthcare professionals that were willing to participate. This included doctors, residents, nurses, pharmacists, biotechnical, and medical advisors. And the MDA analysis was a retrospective study containing reported medical device-related adverse events from previous
properties in which select the according to functional group, thermostables, molecular weight, inert, and pH. The kinetic, the study the inhibition constant. If inhibitors are present in the reaction system, it will help to assess how effectively enzyme can resist inhibitions.
We can detect the early cause of the disease and can give treatment and prevention of disease. Data has also visualized in mobile apps by using electronic health records. And coming to benefits of remote patient monitoring in preventive healthcare, remote patient monitoring has been enabling early detection
of the powdered leaves. The next stage was extraction of the phytoconstituents, which includes plant material for extraction, solvent selection, which I have selected ethanol, methanol, and further maceration process, which I have conducted for 72 hours and repeated the process every day 3 times. And further, I have filtrated and concentrated of my extraction was being obtained through the rotary operator process.
We can able to detect the disease which has been present in that patient, and we can personalizely give the medicine for them in a better way. AI is the best tool for using these medicines in their given way. Challenges such as data privacy concerns, regulatory complexities, and industry-wide adoption barriers remain. However, continuous evaluation of digital capabilities is pharmaceutical landscape. Sir, can you move on the next slide? The introduction here provided for Pharma 4.0 is, it includes different techniques
first esteemed speaker is Ms. Aishwarya Jain, a dedicated pharmaceutical professional and researcher specializing in novel drug delivery systems, nanotechnology, cosmeceuticals, and biomedical applications.
These two are specifically based for the green synthesis methods, and last one, this is basically for chemical and physical methods. So here you can see whenever you prepare the formulation which is in a chemical and physical way, so you can see the final product, or during the whole process, a lot of toxic chemicals are going to be evolved, which will be a health hazard for everyone. But when we talk about green synthesis, what is the starting product? The starting product that we are going to use is bacteria, fungi, plant extracts, and algae, which we do obtain it from the nature, and hence it is free from all the toxins. So this we have seen the innovation in green synthesis. These two methods do produce a lot of toxins, and this method is the best one. We'll talk about it later on. Now, next we'll talk about the importance of green synthesis of metallic nanoparticles in healthcare. So I'll majorly talk about on metallic nanoparticles. So first is minimizing the toxic byproducts in metallic nanoparticle synthesis. So whenever we formulate any formulation by the very common and conventional methods, we will see that lastly the byproduct that we are going to get will be a toxic one. So over here we are going to minimize it because the starting product is natural based. Okay, so when you are using a starting product which is natural, obviously the end product will also be a natural one. So example, we'll see silver nanoparticles synthesized using curcuma longa, that is turmeric extract, exhibit potent antimicrobial activity without the cytotoxic effect associated with the chemical synthesized counterparts. So this is a very common thing when you have started your journey in B pharmacy. The very first thing you must have learned is whenever you take any formulation, there are side effects, right? So when we talk about green synthesized methods, there will be no as such side effects that you're going to observe after you take any formulation. So that is the best part of this. Next is reducing environmental impact in pharmaceutical process. So example, we'll see gold nanoparticles produced from Zydeco indica, that is your neem tree, extract for drug delivery and imaging applications synthesized at room temperature in water, ensuring minimal environmental impact. So this is very important. So whenever you try to synthesize anything, a normal synthesis method, you'll use a lot of organic solvents. And finally, what will happen? There will be increase in temperature. You will see when you use ethanol, alcohol, chloroform into your formulation process. So after that, what you do, you evaporate all these solvents. And these vapors, actually, it is contaminating our air. But when we talk about green synthesis methods, these are used by using water. So because of that, you will see there is minimal environmental hazard created. Lastly, the enhanced biocompatibility and therapeutic efficacy. So magnetite nanoparticles synthesized using Eucalyptus globulus, that is your eucalyptus leaves, extract demonstrate high potential in hyperthermia treatment for cancer with reduced toxicity and enhanced magnetic properties for targeted therapy. So this is very, very crucial. When we talk about when we think about treating any cancer patient, you must have seen a lot of patients undergo chemotherapy. There is hair loss. There is a lot of problems, or there are a lot of side effects which happen with the body. But over here, you will see that the toxicity is largely reduced. So you will see that even if the patients are treated with such kind of formulation, they will not see very deleterious effects onto their body or onto their health. So this becomes a very good, you can say, a better alternative from the conventional formulations. Next, the pharmaceutical applications of green synthesis metallic nano. So we'll see first is targeted drug delivery. So although you might feel that, yeah, it is so easy, we can prepare, anyone can prepare it, then you might feel in the back of the head that, okay, then I think that this formulations won't work as much as better as the conventional ones. So yeah, so let me break your myth over here. So the gold nanoparticles functionalize with anti-cancer drugs for precision oncology treatment. So you're going to get precise treatment even if you synthesize your formulation using green synthesis methods. Then antimicrobial agents. So this is very common that since your childhood, your mom, your dad, your grandparents must have said to you, you should eat food in silver vessels. So why? Because they have antimicrobial activity, or the chawanprash that you eat. So they do also have silver particles present in it, because it has antimicrobial activity. Now, when we know this, like the metals that we use in our day-to-day life are going to have such a wonderful effect, then why should we not incorporate it into our formulations? So this is only achievable with the green synthesis methods. So diagnostic imaging. So until now, we only knew that diagnostic imaging can be done with the synthetic products, but that too, we can do it by using the iron oxide, because iron, it has magnetic properties. So once you insert it into your body, you can use external magnet, and you can target the drug delivery. So the platinum nanoparticles, if you're thinking about anti-cancer therapies, you can also think about platinum nanoparticles. And lastly is the gene delivery. So this is a very crucial part. The above ones were quite basic, but gene delivery is a very crucial aspect or application I can say. So zinc oxide, being a very cheaper metal, can be used for such a humongous or such a deleterious, you can say, health hazard, okay, or a disorder. So rather than talking a lot about this background study, I would really want to go on to the case study so that you will understand how to formulate these, you can say, nanoparticles and how to evaluate those. So this case study will show the procedure, how to prepare, how to evaluate, and what will be the final outcome from this study. Okay, so today I'm going to take a case study in which we are going to prepare zinc oxide nanoparticles by using Terminalia arjuna. So that is called as Arjuna bark. So first of all, what you have to do is you have to take the, you can use anything. You can use leaves, you can use flowers, seeds, you can use roots, barks, algae, anything. Okay, but over here, we'll talk later on. Like over here, I'm going to take a bark. So this bark is loaded with quercetin, gallic acid, and ellagic acid, which are key elements or key biomarkers which have a very good wound healing and antimicrobial activity. So this case study is majorly about proving the potential of this bark and zinc oxide nanoparticles for wound healing activity and antimicrobial activity. So this is the bark that I have taken, which was dried. Then you have to crush the bark and make it into a powder. Later on, you have to macerate it, okay, into an water. After the maceration process, you will filter the extract. So this is the final extract, that is the extract which will be packed with the phytochemicals such as polyphenols, a lot of pollution, and in physical one you use lot of equipments, because of which there is a lot of high energy consumption. So ultimately your final product will be very costlier one. Now when we talk about biological synthesis, in which there is we talk about green synthesis methods. So in this we use a reducing agent. Okay, now what is a reducing agent that we are using? The plant extracts, the seed extracts.
Role in advancing drug safety practices.
Currently serving as a safety risk lead at Payal, she is also a certified psychotherapist and hypnotherapist advocating for mental health. Her scholarly contributions include editorial work with the Journal of Pharmacovigilance and Drug Research.
Today, she will present on pharmacogenomics, the future of personalized medicine. Please join us in extending a warm welcome to Dr. Payal Gajbhiye. Over to you.
Yeah, hi Raheen, hi Bhavika. Thank you so much for introducing me to all the participants. A very good evening, everybody. My name is Dr. Payal Gajbhiye. As you already been introduced about me, I will be taking today the topic of pharmacogenomics. I'd like to present my the drug acts on the cells, the way it is supposed to act on a diseased cell that will get affected, or the enzymes can get triggered and the metabolism can get very fast or very slow. So it will lead to toxicity overdose, or it can get to underdose. So the areas are so broad that finding, that is the reason that there comes an importance of finding the polymorphism and appropriately targeting it. So again, broadly, you see that in current practice, you have a group of patients, you have a doctor. The doctor will prescribe medications. As we saw in the previous slides, patients will either land up having ADRs or serious ADRs, or the 40% people who are not having the intended effect. If you have a preemptive PG pharmacogenomics guided treatment, so if the doctor is actually using the approach of pharmacogenomics guided treatment, what the doctor can do is they can give a modified dose for those where they know that it will be over metabolized or under metabolized, and optimum therapeutic doses can be you can modify the dose to meet those variations. If the drug is too toxic, if on testing you get to know that the drug is will not be suitable, the doctor can give a completely alternative drug. And if no abnormality is detected, then they can go as usual, the treatment, and a normal dose is given. And that is how you can, before prescribing a medication, a PGX guided treatment plan can be made, and the patient can be appropriately categorized, and proper approaches can be taken. Here comes the importance of pharmacogenomic testing, because using this test, you get to know whether a certain medication is going to be effective on you or not. Find out which medication you need. It is possible to predict whether a serious side effect may happen from the medication. So what happens is that the genetic testing, for that the samples are usually taken from blood, saliva, or inside of the cheek, and they are subjected to various pharmacogenetic tests. Although we have not reached a stage where all the drugs have this kind of tests, but still a lot of research has happened where these tests are available, especially like I said for amilocavirine for anti-HIV treatment. Carbamazepine is an epilepsy treatment. For that also it is available. Tamoxifen, warfarin, and clopidogrel. So these are very commonly used drugs, and on them also this test can be done for them. Like there are quite a lot of tests, but I have, for the sake of this presentation, there are few which I have put on the slide, which is like warfarin sensitivity testing. So VK-ROX and CYP2C9 genes, these are the two which are usually involved in its metabolism. And using this testing, we are having a focus on these two genes to find out if there are any abnormalities. This is a very big advancement in current medicine. The KRAS mutation testing for colorectal cancer. So the people who have KRAS mutations have a different approach, and the targeted precision medicine therapy can be used for these patients. Same way for breast cancer, you have HER, human epidermal growth factor receptor testing, which is used for the entire treatment plan of these patients. These are a few of them which are right now very important in clinical practice. Coming to the average costs, so anywhere between $250 to $630. I mean, this is in dollars, but the country to country will vary a little. As per the data in 2022, these are the average costs which I've tried to gather. So what happens is we can see that this looks quite expensive, right? I mean, all this costs. But if you see the effectiveness, if you measure it in terms of the quality of life, that has a significant impact. Like suppose you tell me if a patient is being given an major depressive disorder, a patient is suffering from that, he is at a risk of suicide, right? You are giving antidepressant, it's not working. And the cost to the life of patient is huge, isn't it? Because they may end up harming themselves. Now, if you are able to do a pharmacogenetic testing, you know precisely which drug is going to act on this patient. You are actually going to save his life in ways more effective than money can even cost. So just say the effectiveness is not only about cost, but the quality of life also. And doing a hidden trial on the same patient and not getting results, giving drugs and then not having that efficacy, and then finally zeroing down to a drug that actually works, it ends up losing a lot of treatment time for them. They end up spending a lot of money on the previous medication, also getting frustrated as a result, and their health deteriorates. That is a different matter altogether. So if you see holistically, this entire testing is having a very huge impact on their quality of life, and there is a very dire need that the entire healthcare system should be incorporating these tests during the treatment. Now we are going to look at some of the examples of individual drugs, and now we are going to understand how all these concepts work together in a clinical practice. Clopidogrel, as you all know, is a blood thinner. It's used to prevent cardiac problems like stroke or heart attacks. The active metabolite is converted, the drug's metabolite is converted into liver. What happens? So the liver will break it down. It goes into the blood. It goes and stops the platelet aggregation. And that is how, it prevents strokes. The CYP2C90 is responsible for converting this drug into its active metabolite. Now understand, whenever there is a variation, like we had earlier discussed, if there is a variation And if the C19 poor metabolizers, if it is identified that the patient is a poor metabolizer, it means that they will not make enough active metabolite, right? So it will not have intended effect. So what might happen is the doctor is prescribing the drug. Doctor is not aware that the patient is poor metabolizer. And then, just one second. So then, even if they're prescribing the dosage which they think are appropriate, the patient is actually not having the intended effect, and nobody is probably even aware of it, right? So one fine day, they might land up with stroke and cardiac problems despite taking the medication regularly, despite adhering to the medication at the dose which is prescribed.
Our third speaker of the evening is Mr. Rahul Pal, an accomplished pharmaceutical researcher and assistant professor at ISF College of Pharmacy, Punjab. With an M.Pharm in pharmaceutics from NIMS University and a B.Pharm from Inverness University, he has published over 30 review articles and 11 research papers in internationally recognized journals. His expertise spans drug formulation, computational drug design, and nanotechnology-driven drug delivery systems. Mr. Pal is also a textbook author, patent innovator, and active participant in faculty development programs and symposiums. Today, he will speak on nanotechnology-driven innovations, transforming clinical practice through industry collaboration. Let us welcome Mr. Rahul Pal to the stage. Over to you, sir.
The CRISPR-Cas9, which will use as a majorly for the treatment of the sickle cell anemia for the gene editing or the gene involvement for the particular therapy. So ultimately, there are lots of examples. If I'm talking the industry role in the advancing mineral medicines, so without industry, without ideas, without laboratories work, without the researcher as well involvement, there is no chances for the particular therapeutic or the therapeutic or the treatment. So ultimately, these are the all have the alternative collaboration between to each other. So the next one is the real world case studies. So if I discuss the lots of example, the lots of the theories and the lots or the introduction of the basics of the industry or academia collaborations, is there any actual suitable example for the treatment or as well as the case study for the success story? If I'm talking, the term is a success story, that will be a visualize another thing, just like the heaven of the research field as well. So then key uh, few of the example which we discussed regarding this, the treatment or as well as the case studies or the success stories. So nanotechnology has revolutionized healthcare through the targeted therapy, advanced diagnostic and precision medicines. And it will a collaboration between the pharmaceutical companies, research institutes, technology confirms, and the translating the nanotechnology breakthrough into the real world clinical solutions. And so the first example I am taking lots of time, I am saying that Doxil is the main brand for the liposomal carrier for the treatment of the cancer treatment, which is using of the uh, cancer treatment or using the doxorubicin for the as a main first treatment. So the Doxil is the first FDA approved nanomedicine for the cancer treatment. So if I'm the focusing on the Doxil because it is the first FDA approved medicines. So before that, there is a no liposomal studies also conducted for the particular cancer treatment, but FDA have authorized or giving the permission for the particular marketed formulation availability on the hand, and they will approved for the first nanomedicines for the cancer treatment. And which industries also collaboration, Johnson and Johnson and Elza corporation is a being a most important for the Doxil K productions and then the distribution as well, if I'm talking. And the technology used the liposomal nano carrier. So Doxil is also involving the doxorubicin HCl liposomal injection formulation for the particular bioavailability, for particular targeting of the cancer treatment, for particular the receptor for the cancer as well. So and so why we are using the liposomal? formulation for the cancer therapies. If I'm talking, there is a lots of conventional formulation also available for the marketed formulation. Then will be the ultimately the comparison between the two formulation that enhance the drug delivery directly to the tumor site, and follow their and show their therapeutic effect, and the treatment will be the more curable. And the next one is the improved patient outcomes in terms of ovarian cancers, multiple myeloma, and a lots of S type of cancer, as well as bladder cancer and pancreatic. So there are the lots of industries also collaborated with the nano carrier for the particular targeting or as a success story for the treatment for the particular chronic disorders. Next one already I am discussed, abraxane. So abraxane is also a targeted nanocarrier chemotherapies which involve the Calgene and Abraxis Biosciences industry collaboration between the nanocarrier and the formulations. And the use and the albumin bound या फिर albumin bound nanoparticle for the technology or the treatment chemotherapies. They are also used for the enhance the drug delivery for the breast, lung, and pancreatic cancer as well. So, and then increase the tumor penetration through permeation from the triple B, and improve the viability inside the bloodstream, or as well as the patient compliance reductions, or then it will be more survival of the human being after spreading or after complications with the cancer. So there is a lots of example, or then the treatment as well as the nanotechnology or nano carrier being a most important or a vitally important part of the study. So next one is the mRNA COVID-19 vaccine. I'm talking about Pfizer and biotech Moderna, which we use in the lipid nanocarrier as the formulations. mRNA vaccine against the COVID-19 allowed the rapid vaccine development and there the neutralization of the virus movement of the virus, or as well as the killing after the some time of the formulation. And that will be a demonstrated high efficacy more than a 90% in the prevention of the severe COVID-19 vaccine. So without biotechnology, that will be a not curable, or if I'm talking, the treatment of the COVID-19 was not to be possible, because without the bacteria or a viral or virus structure, that will not be the possibility for the treatment. So ultimately, the biotechnology industries, researchers, academicians, all they have the collaboration between the two each other for a particular targeting or particular treatment. And next one is a nanotherm. Nanotherm is a magnetic nano particle for the cancer therapies, and which using the industry's MagForce AG. Ultimately, the formulation have the wider range or wider treatment of the particular targeting. So they using the super paramagnetic iron oxide nanoparticle SPIONS, which available as a nanotherm for the heat inducing nanoparticle for localized cancer treatment. If we are not destroying the cancerous cell, that will be a more harmful or more spreadable for inside the body or inside the cell as well. So that will be a more complications for the treatment. So ultimately, nanomedicines have the capability or have the advantage or merits for the treatment, for particular viability enhancement, for particular targeting, for reducing the side effect from the conventional dosage form, as if I'm comparing between this. So ultimately the minimal invasive nanomedicines, there are the lots of invasive or non-invasive techniques also involved. So ultimately, if we are using liposomal injection formulation, then we will give the formulation by the IM or IV as well. So ultimately, there is a lots of chances for the treatment within the given periodic table time or the treatment time. So ultimately, the prostate cancer, brain cancer, also the using with the nanotherm as a magnetic field, or as well as the magnetic nanoparticles formulation. And the last one is the nano sponge therapies. So nano sponge is also neutralizing the bacteria, toxins, and virus. So there is a term of MHC. MHC is the major histocompatibility studies or complex studies, in which we will we will generate, or we will study the between how we will completely destroy the virus or bacteria as well, or how will be the neutralize their actions as well. If we are neutralizing, that will be also option for the treatment. And if we are totally destroying the bacteria or virus, then it will be a next another time for killing the bacteria, and for completely destroyed it, we are using the ultra lasers approaches for the treatment. So there we will use the UGS and DIGO and Lightica Therapeutics for the industrial collaboration between the nanospherical formulations, and it will absorb the bacterial toxins, the responsible antigens, pyrogens for the particular infections or virus, or as well as any type of disorder, disease. And that will be preventing tissue damage as well from the sepsis and MRSA infections. There is have then if they having the chrono, if I'm talking the chromosomal disorders. So if we are talking, or if we are previously diagnosed the treatment, that will be a more beneficial for the particular patient or particular therapeutic effect as well. So it will be, and this will be explored the neutralized viral infection, including the COVID-19 is the most vitally if suitable example.
As we come to the conclusion of this enriching symposium, we extend our sincere gratitude to all our esteemed speakers, participants, and organizers for making this event a grand success. Now, I invite Ms. A.V. Vasanti, Vice President of SCRMP Pharmacy Wing Student Forum, to deliver her valedictory address. Over to you, A.V. Vasanti.
A warm good evening, everyone. I'm A.V. Vasanti, the Organizing Secretary of Second National Symposium of SCRMP and Vice President of SCRMP Central Executive Council Student Forum. It's an honor to deliver the vote of thanks for the Second National Symposium of SCRMP with the theme Collaborative Innovations in Healthcare, Bridging Industry, Clinical Practice and Patient Care, which has been truly insightful journey for all of us. The first SCRMP symposium was held in August and September 2024, and today we are proud to be part of the second symposium with the incredible event. We have received an overwhelming responses with more than 70 participants, making this a successful and enriching two-day symposium. On the first day, we had a privilege of listening of two expert sessions from Chinmaya ma'am and Riya ma'am, and on the second day, followed with the witnessing excellent oral presentations from over 20 plus participants, judged by our esteemed judging panel, Gajbhiye sir and Payal ma'am, board members of SCRMP, followed by Shrikant sir, Aishwarya ma'am, and Rahul Paul sir. Thank you so much for the esteemed judging panel for their contribution, which would help the participants in the future endeavors. A heartfelt thank you to the expert speakers for sharing their valuable knowledge and insights with us. So finally, we will also like to express our gratitude to all the participants who have been part of this symposium, contributing with their enthusiasm and hard work in the presentation. I hope each one of you has gained valuable knowledge and experience through this event. A special thanks to our SCRMP board members for providing us with the opportunity to organize this national symposium. Their support has been crucial factor in making this event possible. And a big thank you to the Central Executive Council Student Forum and even the SCRMP State Chapter for their efforts, dedication and teamwork to execute the smooth event from the starting to the end. A big congratulations to the participants and winners for their outstanding presentation in the oral presentation segments. You will receive your exciting rewards shortly. If you wish to explore more about SCRMP and take advantage of incredible benefits, this is the go, where you become a member by visiting the SCRMP website, www.scrmp.in. And if you feel to contact us, please mail us through studentforum.scrmp@gmail.com. Once again, thank you all for your participation. Have a wonderful day ahead.